Thank you. Good evening. My name is Joseph Creanza, and today I will be discussing the scope of essential oils for the control of soybean white mold disease. So in today's agroecosystems, several factors stipulate the success of a given crop. Currently, one of those factors, plant diseases, causes anywhere from 20 to 80% of crop loss in the field, with an additional roughly 10% loss due to post-harvest diseases. Now, up to this point, chemical control of diseases has been the most common management strategy, but this comes at an extreme cost. Wisconsin farmers invest roughly $23 million annually for the control of these diseases. Not to mention the economic impact, the development and use of synthetic fungicides cause detriment to the environment, as well as promote the development of fungicide-resistant pathogens. In the Midwest, white mold prevails as the dominant plant disease affecting soybeans. Now, essential oils have been used for thousands of years for various different purposes, including their role as antimicrobial agents. And testing the scope of essential oils as antifungal agents for the control of soybean white mold disease is a step for sustainable plant disease management in tomorrow's agriculture. So before the study was initiated, we decided to break our approach down into 3 stages. We began by identifying a concentration of essential oil that inhibited fungal growth on the plate through a growth inhibition assay. This starting point gave us an idea of the smallest concentration that would have an effect on inhibiting mold growth on the plate. Simultaneously, we initiated a phytotoxicity assay, where we identify different concentrations of essential oils with the least phytotoxic effect. We did this to prevent plant detriment at the time of the oil application. Lastly, we aim to identify an essential oil that works best in a control and preventative strategy through a pathogenicity assay. To identify a concentration of essential oil that inhibits white mold growth, we utilized and initiated our study with a growth inhibition assay. This experiment utilizes a strategy known as poison food technique. We started by preparing fungal growth medium plates and treating them with 6 different essential oil treatments. White mold was then subcultured onto the treated plates and left in incubation for a week's time. As you can see on the pictures of the right, there are 2 different essential oil concentration treated plates, and you can see that there is even no mold growth even at 0.075% concentration. Our second step in the study was to identify concentrations of essential oil with the least phytotoxicity. We did this by utilizing a phytotoxicity assay in which we used 5 different essential oil concentrations to test the effects on soybean foliage. The soybeans were sprayed with different concentrations, left for 3 days, and then the phytotoxicity severity was scored. As you can see from the pictures, there are different phytotoxic effects for different concentrations of a particular essential oil. As you can also see, 0.075% concentration proved to be the least phytotoxic, in addition to also being effective in inhibiting white mold growth on the plate. Our final approach, to identify an essential oil that worked best in a control or preventative strategy, was utilized through a pathogenicity assay. Here, soybean seedling infection severity was scored in 2 different management practices. The first group of soybeans were inoculated with white mold first, rested for 3 days, then treated with essential oil to test the effects in a control strategy. The plants were then sitting for a week's time, and the disease severity was scored on the 7th day. The second group of soybeans were treated with essential oil first, then inoculated 3 days later to test the effects in a preventative situation. The plants also sat for a week's time, and the disease severity was scored on the 7th day. As you can see, the disease severity scores were significantly lower in the soybean seedlings that were treated under the prevention strategy comparative to that of the control strategy. So from these three approaches, we found several valuable pieces of information that can be utilized moving forward. Firstly, from the growth inhibition assay, we've learned that all 6 treatments have a growth inhibiting effect on the media at as low as 0.075% concentration. From the phytotoxicity assay, we learned that 0.75% essential oil concentration caused the least phytotoxicity to the soybean seedlings. Combining these results, we then tested the effects of the oils in a control and preventative strategy through a pathogenicity assay and found a significant reduction in disease severity in plants tested with a preventative strategy. Moving forward with the project, testing lower concentration as well as more essential oils, eventually modifying essential oil compounds to have improved solubility and efficacy in plants, and gene expression study to identify the defense mechanisms triggered are all of relevance to our study. Essential oils have been used for thousands of years and now may be potentially used as a safe alternative to chemical fungicides in the control of plant diseases for a more sustainable future. Thank you, WiSys Foundation, for holding this virtual symposium in which I was able to present my research, along with my faculty advisor and friend, Dr. Muthu Venkateshwaran, my colleagues at Muthu Lab, and the Summer Undergraduate Scholarship Program at the University of Wisconsin-Platteville for giving me the opportunity to conduct my research this summer. Thank you.